All right, everybody, so we're back again for our module number 9. So all of this week we'll be covering module 9, and that's going to be all about taxonomy and systemics. So you can find your readings for this module in your book in the top left or right corners under pages 147 through 165. So that's going to be your reading and what our next quiz is going to be over. We're going to be covering part 1 today. We'll be covering part 2 on Wednesday and part 3 on Friday. So before I go ahead and get started, I just wanted to remind you all of what is going to be due this week. So we have our Module 8 quiz that's due Tuesday, April 7th. We have those 10 questions. It's going to be open book, 20-minute time limit. That's all about our speciation and extinction. So everything that we covered last week, all of those recorded lectures on our YouTube page, make sure you look at those and be sure to get that quiz done, I believe by 11:59 p.m. on Tuesday, April 7th. So get that turned in. We also have our lab number 8, which is our island biogeography lab, all about the lizards and the phylogeny of the lizards and the distance of how far the islands are, all of that stuff determining that biogeography for those island lizards is going to be due on Tuesday, April 7th. So make sure you get that lab turned in. You have a couple options for turning it in. If you can't scan it and submit it to my email or Dropbox, you can also complete it on the computer. That's another option, and then submit it to me that way. Or if those two options don't work, you're more than welcome to take a photo of each of the pages of the lab. Make sure you submit the pages in order. That makes it a little bit easier for me. And I did get some questions about the phylogeny. Some of you had issues doing the phylogeny on the computer or on Microsoft Word. And if that's the case, you're more than welcome to draw out the phylogeny and take a photo of it and submit it to me that way. So that's another option as well. So get that turned in by 11:59, April 7th, Tuesday, tomorrow. The third thing that we have due this week is our bio news file number 3. So that's our last bio news file that's due. It's due Tuesday, April 7th, just like everything else. And make sure you all are completing it adequately by following the prompts, making sure you're including the works cited page, making sure you're summarizing the article and picking a relevant article if you haven't done it. And luckily it's the last one, so I know a lot of you were not huge fans of the bio news files, but it's the last one, so make sure you get that turned in. And you could submit that to me through Dropbox or email, whichever you prefer, but get it turned in so you don't lose easy points. And then our last bullet point here is your topic presentations or your presentation topics. So for your PowerPoint presentation that's going to be due, I believe on April 21st, make sure you get those topics in by April 10th. And so you should be thinking about something you want to present on. And I've gotten a few responses, but not very many yet. But that is due April 10th, so pick something you are excited to talk about for 3 to 5 minutes. And then what I would like you to do is post your preferred topic under the discussion thread in module number 8 in your Cobra. So go to Cobra, click on your module 8, and there should be a link in the content area there that says PowerPoint discussion thread, and that is where you're going to post your topic. And the reason for that is because I want to make sure you all are doing different topics. So I want to make sure there's no duplicated topics. So get that turned in, first come, first serve on the topic choice. So if there's something you really want to do, get that into me. So that is going to be due April 10th. Okay, so I'm just going to talk about the new assignments that are going to be due. So we have our module number 9 quiz. That's going to be a week from this week. So that's going to be due by Tuesday, April 14th. Everything that we covered this week is going to be on that quiz, and that's going to be open book, 20 minutes, 10 random questions, and that's going to be next week, Tuesday, April 14th. So don't worry about that quite yet. Just make sure you're listening to all of the lectures and you're doing the lab and you're preparing for that Module 9 quiz over the course of the week. Next up here we have our lab number 9 for our communicules. So make sure you are paying attention to the lab. I've posted the lab guidelines already on YouTube. It's about 15 minutes long for lab number 9. You should have that lab in your lab book packet. It's also in Cobra if you wanted to print it out or if you already pitched your lab book. But lab number 9 is going to be due next Tuesday, April 14th. So that will be due then as well. And then lastly, the third assignment that I have for you guys is an Evolving Switches video. So it's a 15-minute video. You can watch it right online. It's free. Only 15 minutes, and there's 9 questions. It's a quiz that's open book. There's no time limit on it. But it is a lot of fill-in-the-blank answers. And so it'll be all about stickleback fish, and that's due Tuesday, April 14th, along with these other 2 items here. So make sure you're doing that as well. It'll really prep us for the week following this week when we start talking about sticklebacks for one of our labs that we'll be doing. So make sure you complete that Evolving Switches video and quiz. All right. So now that I've talked all about everything that we have due, now let's get into our lecture for today. So for Module 9, as I mentioned before, the reading is page 147 to 165. So make sure you're doing your reading in your book to prepare for that module quiz. But we have 5 different sections in that module: taxonomy, phylogenetic trees, cladistics, molecular clocks, and horizontal gene transfer. And today we're going to be primarily covering taxonomy and phylogenetic trees. Now I know we did cover a bit of this in one of our earlier modules, so it will be a little bit of review, but we're going to go into more depth and more terminology regarding this general top subject matter. All right, so let's get started with our taxonomy and systematics. So when we talk about taxonomy, basically this is the ordering of organisms. Now consider the Asian elephant and the African elephant. So for many, many years, scientists only had 2 different kinds of elephant when they were looking at taxonomy of elephants. There was only the Asian elephant and only the African elephant. However, during the ivory trade, the ivory tusk trade, scientists were taking samples of elephant tusks to better track where ivory was coming from, because Asian elephants do have ivory, although their tusks are a little bit smaller and they're mostly only present in male Asian elephants. The African elephants do have tusks as well, but ivory trade is like a huge deal and it causes these animals to die. And so when scientists were trying to track where ivory was coming from, genetically they actually determined that there is in fact potentially a 3rd species of elephant, which is on the far right here. So this is the African forest elephant, and so it's a little bit different. It doesn't live in the plains and in the savanna. It lives in Central and Western Africa, more in the forest and jungles. And so they were forced to redo the taxonomy for these elephant species and classify them differently. So that's just a little fun tidbit that's interesting to consider when talking about taxonomy and getting into systematics. Okay, so let's do some terminology here. So you've probably heard me mention the word taxonomy before, but let's do some definitions. So taxonomy technically speaking, is the science of describing, naming, and classifying these living and extinct organisms and viruses and other creatures. And so describing them, how they look physically, giving them all a name, and classifying them depending on their evolution or who they're most closely related to is what taxonomy is in a nutshell. When we talk about systematics, that is the study of biological diversity and evolutionary relationships among organisms, both extinct and modern, so those organisms that are still living. So we're studying all of that diversity, all of the relationships that exist between those organisms, where taxonomy is more of classifying those living things, naming them, describing them. Now, when we come to taxonomy, basically taxonomic groups are based upon the hypothesis regarding all evolutionary relationships are derived from systematics. So that's how those taxonomic groups are formed or based on those relationships that have come from systematics. So make sure you keep that in mind. Okay, so when we're talking about taxonomy, it's really a hierarchical system, and it involves different levels, these successive levels. And each group at these levels, or at any level, is called a taxon. And so if you're referring to a group that's in a class or an order or a kingdom, that is a specific taxon that we're talking about. So it can be any of those. Now, when we talk about this hierarchical system, the highest level that we're talking about is domain, which is the overarching umbrella, the largest domain that encompasses all of Earth's living creatures. So all of life belongs in one of three domains that we have. So we have bacteria, archaea, and eukarya. So all of these three domains exist, and we belong under one of these domains, which is the eukarya. If you look at the fox here above me in this image here, you can see how it's classified under which domain it's located under, which kingdom, the Animalia, the phylum, which is Chordata, which means backbone or spinal cord, the class, the order, the family, the genus, and the most specific is the species. So that has your specific epithet, which, if you recall from the last module, we'll talk about again today. Okay, in the past, remember we talked about the three domains, bacteria, archaea, and eukarya. So we have those three domains, but in the past, there have been eukaryotic subgroups. So under the eukarya domain, scientists had organized these subgroups. And in the past, which is now considered wrong, were four of them. So we had the protists or the protista, the fungi, the plants or the plantae, and the animals under Animalia. And so those were traditional eukaryotic groups that they had under that domain. Nowadays, unfortunately, or fortunately, they have divided it even larger into 7 subgroups. And so now we have these general eukaryotic kingdoms, Discoba, land plants and relatives of land plants, Alveolata, Stremenophila, Rhizaria, Amoebozoas, and lastly Opisthokontas, which are the fungi and the Animalia. And so now scientists have determined that these 7 groups these 7 subgroups are more likely and that work a little bit better in order to classify these organisms. So when we talk about classification, we have to talk about it in a hierarchical sense, as I mentioned before. So it goes from very broad and encompassing to very specific and distinct. So when we talk about domain, that is the largest group. So in this specific incidence here, we're looking at the gray wolf. So this specific species has been classified all the way down to the specific epithet or the species. So we have our domain. It's in the eukarya domain. We have the supergroup Opisthokonta, which is all the fungi and the Animalia. Then we have kingdom, so it's classified even further. Under kingdom, it is Animalia, it's not a fungi. Then we have the phylum, which is our chordata, which is our backbone or spinal cord. The class, which is mammals, so we have all of our mammals, of hair-bearing, milk-producing mammals in the Mammalia class. And then we have our order, which goes down even further. And this one is carnivora. So we have our carnivores, which is bears, big cats, hyenas, wolves, any sort of carnivore that we have. And then going down even further and smaller into classifying it, we have our family, which is Canidae, which are the dog species or wolf species. So any sort of canine we have in our family. So the wolf, the gray wolf, belongs in our canine family. And then as we go down further, we have our genus, which is our Canis, which is even more distinct in the type of canine that we have. And then lastly, we have our species, which is Lupus, which is our gray wolf species. So when we talk about gray wolf, the proper way to discuss it would be using the genus and the species or the specific epithet, which would be Canis lupus, which is our gray wolf species. So you can see, as scientists have to classify all of these organisms, it goes through a series that really puts it into little boxes to help it become more distinct and to help scientists best describe a specific species. Now, if you all remember, we talked a little bit about binomial nomenclature in the past. And we're going to talk about it again just because it is really important when we're talking about species. Now, our binomial nomenclature is the naming system that Linnaeus gave to species that he described years and years ago. And in order to use that binomial nomenclature, it consists of two words, the genus name and the species epithet, or it's a fancy way of saying a specific species. So when we talk about humans, we ourselves are Homo sapiens. And so that is our genus name and our species epithet. So the genus name, in this case, Homo, for Homo sapiens, the genus name is always capitalized with a capital letter. So Homo has a capital H in it for the very first letter. Now, for the second word here, our species epithet, that specific word, sapiens, in this case, is never capitalized. And both names are either italicized, but if you're writing it out and you cannot italicize the genus and the specific epithet, then you would therefore underline the words. So that is only if you were handwriting it. Now, if you're typing it on the computer, you're able to italicize the words, so make sure you do that. And so these are the rules that we use for that have been established and that are regulated through many international associations. So it's used all across the world, and it really does help distinguish which specific organism we're talking about. And so this is why it's so important, because there's a lot of different names for a lot of different things. And when we use the genus name and the species epithet, it really helps us to determine what we are in fact talking about. Okay, so I have here a short video with the Amoeba Sisters. So hopefully we're going to try it and see if this works. I hope it does. I don't know. I've never played a video inside of a video before. So we'll see what happens, and we're going to hope for the best. I'll still be here, But you'll see the video up in this corner here, and hopefully you'll be able to hear it, but we'll see.
There are a lot of animals that I have always thought are cool. Salmon, hairless guinea pigs, iguanas, silky bantam chickens. But one day in my 7th grade science class, my teacher told us that we would get to see one of the most amazing animals ever in class, a hydra. And I have to confess, what I imagined in my head and what was actually reality was a little different, but I'm kind of used to that. Turns out hydra are pretty cool. Hydra are animals that are very small, a few millimeters in length, and they live in fresh water. They can viciously attack and eat their tiny swimming prey, and they can reproduce by budding an identical offspring off of themselves. But if you saw them, you might not think they're an animal. What makes us categorize them as an animal anyway, and not a plant or a protist or a fungus? In fact, how do we classify in the first place? Well, this lets us jump right into our topic, classification. Taxonomy includes the naming and classification of species, and much of the credit for starting a formal classification goes to Carl Linnaeus. We'll get back to that a little bit later in this video. But back in his time, the 18th century, there wasn't a separate category for other organisms like protists or bacteria. They didn't know about detailed cell structure differences. They didn't know about DNA, so they didn't know genetic relationships that now can determine how we categorize organisms. And that's the thing about classification. As we learn more about DNA, and therefore relatedness, how we classify organisms can continue to change. So first, let's look at an awesome hierarchy system mnemonic. There's a lot of great ones that you can find that people have shared online. We'd also love to share ours. Dear King Paramecium cares only for green spirulina, but we sort of have a thing for protists, and we realize that might not be as memorable. So let's take a look at each group of this hierarchy, starting with one of the most inclusive groups, domains. Domains. It's so awesome that all of life will fit into them. So there are 3 domains, bacteria, archaea, and eukarya. The domain bacteria is full of bacteria. They're prokaryotes, and therefore they have characteristics we've mentioned of prokaryotes before. These can include bacteria that make you sick, the bacteria that are in your intestines helping you digest, the bacteria helping with decomposing, the bacteria fixing nitrogen in the soil, tons of different kinds of bacteria. Archaea are prokaryotes too, but they have some major DNA and structure differences that give them their own domain. And while they may seem more closely related to bacteria since they are prokaryotes, recent DNA evidence links them to having more in common with eukaryotes, which is interesting. Many of the archaea are extremophiles. That is, many of them like the extremes. Some like extreme salt environments, for example, which means they can handle extremely salty environments like the Dead Sea. Or methanogens. They can live where there is very little oxygen. In fact, most of them can't even handle oxygen. They use carbon dioxide to make their energy instead and produce waste gas, methane. Some of them live inside animals that eat a lot of cellulose, like cows or termites. Another extremophile is thermophiles. They like extreme temperatures. If you ever dreamed of living near the deep sea hydrothermal vents, well, if you were a thermophile, you'd be in luck. We mentioned that archaea and bacteria domains are separate because they have some major DNA and structure differences that are significant enough to separate them. And so does the third domain, eukarya. These are eukaryotes, and so they have characteristics that we've mentioned before that are common for eukaryotes. And that's where we'll focus right now. So the next level, less inclusive and more specific than domains, is the level of kingdom. Now here is the big disclaimer about kingdoms. Each organization is often changing, and it's not even something that all scientists agree on. We've seen a 5-kingdom system that looks like this, and a 6-kingdom system that looks like this, and it's important to understand it's a changing view as we learn more about DNA and cell structure evidence. That's what makes classification so exciting. But if we focus on these eukaryotes, let's touch on them briefly. Protista, extremely diverse, and there's often talk about dividing it because of how diverse it is. There are protists that are animal-like, and protists that are plant-like, and protists that are fungi-like, but many scientists don't consider them to quite meet the requirements to be in those kingdoms. Protista includes both autotroph protists, making their own food, and heterotroph protists, which can consume other things for energy. Most protists are unicellular, but they can be multicellular. And some have cell walls made of cellulose, which is kind of similar to plants, and some don't. Fungi are heterotrophs, so no photosynthesis for them. If that's hard to remember, just think about athlete's foot. It's a fungus on your foot, and it's not doing any photosynthesis there because that'd be really weird. Nope, it's there, causing irritation, eating dead skin cells. Fungi are usually multicellular, but they can be unicellular. And most of them have cell walls of chitin. It's a carbohydrate. Next up, plantae. Plants are autotrophs. Yes, even the carnivorous plants, because they still make their glucose from sunlight energy. Plants are, for the most part, multicellular, and most have cell walls of cellulose. Finally, last up, Animalia. Animals. Okay, Hydra, you can come back. This mostly multicellular and heterotroph kingdom is the kingdom to which you belong. So now we have the other hierarchy levels. We get less inclusive, therefore more specific, as we move down to the Hydra's phylum, class, order, family, genus, and species. Because the species name is the most specific that you can get, it is the least inclusive. Now, you'll notice we wrote its species name here. Remember how we said we'd bring up Carl Linnaeus again? Well, it's because of him that we have this naming system, binomial nomenclature. This two-part naming system that we uses Latin or Greek roots. This is its scientific name. See that first name? That's its genus. It's written with a capital letter at the start, and it's written in italics. See the second name? That's its specific epithet, which is a fancy way of saying that it refers to one species in the genus. It has a lowercase letter, and it's also written in italics. So why do we care about these scientific names? Well, you could come up with a lot of common names for an organism that vary based on location. Take this mountain lion, for example. It's also known as a puma, cougar, or Texas panther. Same animal, different names. But its scientific name here is specific and recognized regardless of your location. And that gives power to an awesome way to organize and name species. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Alright, so that was a great example of some reasons that we have binomial nomenclature. Also went into a little bit more detail on protists, fungi, and Animalia, and plant species. So I thought that was a good little video. Hopefully it works in this recording, but if not, then we'll figure something else out. Alright, so let's get into phylogenetic trees. So this is the second portion of the second topic that we're covering today. So phylogeny, basically. So let's do some more definition time. So our phylogeny is basically the evolutionary history of a species or of a group of species. So this is going back, what is its history throughout time? Who is its common ancestor? How did it evolve through to be what it is today? So that is our phylogeny. Now, in order to propose a phylogeny, biologists use the tools of systematics. So they're using this specific tool in order to propose the evolutionary history of a specific organism. And so, for example, trees, when we classify a tree, it's usually based on morphological or genetic data. So it's looking at the genes, it's looking at the genetic data, and also the physical makeup of the tree or the morphological data. So when we talk about phylogenetic trees, it's basically what it is in short is it's a diagram that describes the phylogeny or the evolutionary history. So it's a diagram you can actually physically look at to see, where is a specific organism evolving from. Now, basically, when we talk about phylogeny, it is a hypothesis of evolutionary relationships among various species. So it's this hypothesis of how a certain species evolved, or the relationships of a species. And we don't know everything exactly for sure, but it's based on available information, what we do have. Now, when we are looking at new species or species that haven't been described before, we have new species that can be formed by one of two ways. We have anagenesis and we have cladogenesis. When we talk about anagenesis, this is a single species that is evolving into a different species. So say you have a flower that is evolving into something that looks like a flower but is not quite the same as it was, but it's coming from that original flower. When we talk about cladogenesis, this is when we have a species that diverges into two or more species. And so we have one species that branches off into two and then is now two separate different species from what the original was. And we'll talk about anagenesis and cladogenesis again when we look at our phylogenetic trees. And so you can see here's an example of a phylogenetic tree. Up above me here, you can see we're talking about lamprey, tuna, salamander, turtles, leopards, and how they are positioned based on evolutionary history with a vertebrae, hinged jaws, 4 walking legs, amniotic eggs, and hair. And I think I had something similar to this in our last module as well. Okay, so here's a really good example of a phylogenetic tree, and we're going to walk through this step by step just to talk about each point and to show how a species can change over time, how it can evolve, how it can change, and then how this phylogenetic tree can help us describe those changes. Okay, so let's start at the very beginning. So over here we can see at the very bottom of our phylogenetic tree we have species A, and so this species here of this flower, this is where it begins. And so when we have this original species that exists, this yellow type flower, eventually it's going to branch out into, it's going to change into two separate species. So this is our branch point or our node. And this is going to indicate when a species changes into two or more species. And the node is that divergence, that change from this one straight line into two or more different sections. And so let's go through on the right side first. So the right side of your screen. So from species A, we end up developing into species B. And so over time, we have species B that comes from species A. And once we have B, then we have something that's occurring here. When we have species A, it's diverging into species A and B. So this is called cladogenesis. Remember what we talked about on the last slide here? Cladogenesis is the species diverging into two or more species. And so that cladogenesis is when that species A is becoming species A on the left side and species B on the right side. So this is our cladogenesis happening. So now we have species B on the right side. We're still on the right side. We're going through this now. So after we have this divergence into a different species, into A and B here, we have the next step in the phylogenetic tree here. And so the species B here we have goes to the left and to the right. And so the group on the left, you have another node, but that node continues to include species B. So you do have species B over time, but you also have species E that is changing at the tip of the branch. And now you can see, if you look on the far left, we're looking at the change over time. So this is millions of years ago. And so time is going from the bottom of the phylogenetic tree all the way to the top of the phylogenetic tree. So when you have the species B of flower, It evolves into species A, but you also have species B as well. But you can see where it stops on the diagram. It stops below where present time is. And so therefore you can say that the species at the tips of these branches here, based on where they're positioned, that these species are extinct species. Now, if you look on the right side here, species B evolves into species D, or changes into species D. So this group includes all the species that were derived from the ancestor species D. So you can see after it goes into D, it comes out with J, H, and K. So D eventually gives way to species J, H, and K. So this is what we would call a clade. And so this clade is this group that includes all of the species that were derived from species D. So if you were to put a big circle around J, H, and K, and D, that is what we would call a clade because it has all of the ancestors and for that specific group and all of the descendants that it had derived from. Or all of the descendants that derived from the ancestors. Pardon me. So if you go back, let's go back onto the left side of the diagram here. So now we have species A that hit a node and then became species C. And then also on the other side here we have F and I. So eventually A became species F and I. And it also became species C, but C over time changed into species G. So this is what we would call anagenesis. This is where a species, in this case, species C, evolved into species G. So it was C, now it's G, it's changed a little bit. But you can see also in this section of the diagram here that F, I, and G is also alive and during present day. And so you have species F, I, G, J, H, and K at the very tips of the branches of this phylogenetic tree, and they are present and they still exist today in today's time. And so this is just like kind of a way to look at a basic phylogenetic tree where we have most of our terms here with anagenesis, cladogenesis, our clades. And we can see how it has changed over time. We have our nodes where our diverging points. And this is just a basic phylogenetic tree, just to get you guys more accustomed to what they would look like. Okay, so here we have another phylogenetic tree. So this diagram, it has the same kind of setup with time going from the bottom of the diagram all the way to the top. So at the bottom it's very old, 500 billion years ago or million years ago. And then we have time all the way up to the present. And so you can see that species number 43, this is showing us the relationship between a phylogenetic tree and taxonomy. So this occurs when taxonomy is correctly based in evolutionary relationships. And so species 43 here, you can see that it has correctly been labeled with the genus, the family, the order, the class, the phylum, and the species. And so the taxonomy is labeled here through this phylogenetic tree, and it's based on these evolutionary relationships. And so this is what it would look like if we were looking at both taxonomy and phylogeny. Okay, so when we talk about phylogenetic trees, there's a couple other terms that I want you all to be familiar with. So the first term is our monophyletic group, or our clade. So this is a group of species, or a taxon, that consists of the most recent common ancestor and all of its ancestors. So this is basically looking at the ancestor and all the ancestors that came from that. So smaller and more recent clades are basically going to be nested within larger clades that have some older common ancestors here. When we look at the next term here, we have paraphyletic groups. And this is the opposite. So this contains a common ancestor and some, but not all of its descendants. So it's missing a group. So we're missing someone in its descendant line. We're not including everybody that should be included, basically. So someone is missing here. So that's our paraphyletic group. And then the third term here is we have our polyphyletic group. Sorry I didn't have it on the last slide, but I have it on this slide here. So you can see right above me on letter C, this is our polyphyletic group. So this is a group that contains groups of species with different common ancestors. So it contains common ancestors of a group, but also somebody that doesn't quite belong and doesn't have the same common ancestor. The common ancestors are different. And so let's go through all three in this form here, because this really helps visualize what we were talking about in the last slide. So letter A, our monophyletic group. This is the common ancestor, all of its descendants or all of its ancestors here. So we're looking at L, M, N, and O letters. So those are all the descendants, and they have ancestors F and G, and they all share a common ancestor C. So that's our monophyletic group. We have our paraphyletic group. So this is the group that contains the common ancestor, but not all of its descendants. So the same letters here, we have L, M, N, but we are not including O. We're missing this descendant. We still have F and G, and then we still have the common ancestor C. But you can see the one difference in this particular image is that we're not including O as one of the descendants. And then we just talked about the polyphyletic group. It includes the groups of species with different common ancestors. So we have L, M, N, and O, F and G, and then C as the common ancestor, but we're also including J and K, which has a completely different common ancestor. That common ancestor is common ancestor B, and so technically they don't all have the same common ancestor when we're circling these particular groupings. So keep those definitions in mind because they will appear again. Okay, so when we talk about our phylogenetic trees again here, over time our taxonomic groups will be reorganized. So only our monophyletic groups are going to be recognized. So those groups that contain all of the ancestor or all of the descendants and the common ancestor. So here you can see we have reptiles, and there's two different sets of phylogenetic trees here. The first phylogenetic tree does not include birds, whereas the second phylogenetic tree does include the birds. So here's a question for you. Reptiles were a blank group because birds were excluded. And that's the first phylogenetic tree that I'm referencing here, is the group that does not have the birds circled. So that's going to be our paraphyletic group because those birds were excluded when we were circling the descendants in this phylogenetic tree here. And here we are again, reptiles as a paraphyletic taxon on the left, reptiles as a monophyletic taxon on the right. So it's including all of the descendants and the common ancestors, whereas paraphyletic on the left does not include the birds, so therefore does not make it a monophyletic taxon. All right, so I have one more video for you and then a few more slides, and then we'll wrap it up for today's lecture. So this is only about 3 minutes long. It talks a little bit more about phylogenetic trees, and hopefully it'll help clarify some things for you.
A phylogenetic tree is a visual representation of the relatedness of species by descent from a common ancestor. The branch tips represent existing species. The branches connect the common ancestor, and the nodes depict where one species diverged into two or more species. Because the fossil record is incomplete and we do not have direct knowledge of evolutionary history, a phylogenetic tree must be inferred indirectly from data. For example, homologous structures, those that are derived from the same body part in a common ancestor, can be examined. The forelimbs of many animals have similar bone patterns. The feathers of modern birds are homologous with feathers found in fossils. In contrast, wings of birds and dragonflies are analogous. They are derived from different ancestral structures. The cornerstone of tree building is to identify shared derived traits accurately. Species that share derived characters belong to a clade. A derived character shared by clade members is called a synapomorphy of that clade. One of the most basic ways to distinguish between ancestral and derived traits is by outgroup analysis. In this approach, traits of the group being studied, the in-group, are compared to traits of a very close relative that clearly branched off earlier, the outgroup. A cladogram is a phylogenetic tree inferred by clustering synapomorphies. In general, cladograms only indicate the order of evolutionary branching events, not the timing of these events. How do we decide whether a tree is more or less likely to be correct? Shared derived characters may have evolved independently as the result of convergence, or may be lost within a group as the result of reversal. For example, adult frogs do not have tails, a trait that unites gorillas and humans. Grouping frogs with salamanders is favored because it requires loss of only one trait, the tail. A phylogeny in which frogs are most closely related to gorillas and humans requires loss of tail, loss of hair, and gain of amniotic membranes in frogs. In the 1970s, researchers began to use molecular characteristics such as comparisons of DNA, ribosomal RNA, and protein subunit sequences to construct phylogenetic trees. These molecular methods have revolutionized phylogenetics. For example, living organisms are now viewed as being divided into three domains: bacteria, archaea, and eukarya. Molecular methods have resulted in a dramatic rearrangement of the protists. Although the fungi are recognized as a kingdom, the algae are now viewed as being scattered among various groups of protozoa. Red algae appear to represent a distinct group, while the green algae are closely related to plants, and the brown algae appear to be related to a specific group of water molds.
All right. Sorry, I had to step away for a second. Oh, all right. So next on our list to talk about, we have our homology. Now, if you remember, we talked about homology in the past. These are basically our similarities among various species that occur because they have a common ancestor. So they have an ancestor in which they've evolved from, and the reason that they have a specific trait is because that common ancestor had that trait, and we have formed from that common ancestor, and so we all have similar or traits that have been derived from each other. So, for example, we have the bat wing, the human arm, the cat front leg, all have the same bones, all have the same ancestral characteristics that have been derived from the same common ancestor. And so we have genes that can also be homologous if they are derived from the same ancestral genes. So even in our genes, not even in our actual features that we have, but our genes as well can be homologous. So if you remember homology, we're just going over it again just to really drive that point home. So if you remember our analogy, our analogous traits here. So these are similarities among organisms that are derived from common usage or adaptation. So these are the features or the characteristics that are being evolved separately and that do not share a common ancestor. So in this specific incidence here, we have insects. Maybe you have an ant leg and also a horse leg. Obviously, they don't have the same common ancestors here, but they are analogous structures. So they are similar, used for similar function, similar form. Same thing with fins, the sculpin fin in a fish, the dolphin fin in a mammal, used for the same form and function, but totally derived from different common ancestors. And then lastly here you have the insect wing. Same thing with the chicken wing. So you have, or a bird in general, you have wings that are used for the same things but have a different common ancestor. So the adaptation is formed completely separate based on the fact that they need it for a particular purpose. Okay, so we also have morphological analysis. So when we talk about morphological analysis, this is basically the first systematic studies were focused on morphological features of extinct and modern species. So this is the studies that were formed first were looking at actual physical traits, morphological features here, physical traits that organisms had. And so that was the very first studies. Now we do some genetic studies as well, in addition to the morphological, and we look at geographical and geological things as well. So when we talk about these morphological features, it's important to always mention convergent evolution. This is when those analogous traits are arising independently due to adaptation, similar environments, and those can cause problems for folks who are trying to classify organisms, because you want to make sure that they're arising from the same common ancestor, but sometimes that can be difficult to determine what is convergent evolution and what is not. So when we talk about molecular systematics here, this is when we're using genetic information to help us classify organisms. So when you analyze the genetic data, such as either DNA or amino acid sequences, to help identify and study those genetic homologies. And so this will help us create that phylogenetic tree that is the most accurate. And so DNA and amino acid sequences from closely related species are more similar than sequences of more distantly related species. So as you'll do your next lab with the communicules, you'll well, I guess no, you did that with your island lizards. The island lizards helped you to determine who was more closely related by counting the number of differences in nucleotides. So that helped you determine who was more closely related. So in this particular case here, you have the elephant shrew on the left, this cute little guy with the big eyeballs. So the taxonomists mistakenly organized the elephant shrew and placed them with the other shrews. However, this was incorrect. As you can see, the other shrews are a little bit different, the ones that you might see in North America here. And so this was corrected, and eventually the elephant shrew was moved to the Afrotheria, which is animals that originated in Africa and not necessarily in other places. So eventually it was corrected and the shrew was classified properly, because this could have happened through some sort of convergent evolution or something in a similar respect. So I think that's all that I have for you guys today for lecture. So I will get the next lectures up and posted here shortly. And thank you all for listening.